So welcome back guys, welcome back to our another video tutorial. So today we are here to going to talk about a topic which is very interesting, and it is belongs to immunology. Briefly speaking, it is a topic of cancer immunotherapy, right? So we are here going to talk about how monoclonal antibody can be used to treat cancer, right? So if before entering to the topic, let's say about what is monoclonal antibody. Monoclonal antibody is a kind of antibody which we have to generally prepare artificially. Sometimes they also have they also prepared naturally. So monoclonal antibodies are antibodies that have the ability to recognize the specific epitopes of the antigen, and they are coming from a single B cell lineage, right? That means it is generally used in clinical and research purpose, where we are interested to isolate a specific antigen from a specific antigen mixture, right? For that purpose we are going to made a monoclonal antibody by fusioning melanoma cells and activating antibody producing B cells, right? And now why we need to fuse a B cell with a melanoma cell? Because if we not add B cell with melanoma cell, then this monoclonal antibody will not going to survive for a longer time, and that will be a loss. And we all know that melanoma cell, that is our cancer cell, are rapidly dividing and rapidly growing. That is why they are fused with B cell to produce monoclonal antibody via hybridoma technology. So although we in case of treatment, cancer treatment, on the aspect of cancer immunotherapy, we are generally interested to augment our own immune response against the cancer, right? By simply enhancing our APC activity by culturing dendritic cell with GMCF, so that it will activate our helper T cell, and it will in turn activate humoral immunity as well as cell mediated immunity. We have also apply B7 ligand in the melanoma cell, so that it will it can express the B7 ligand, which will B7 proteins, which will have the ability to provide the co-stimulatory signal. In that way, we generally focusing on augment our own immune response to fight against cancer, right? It is, and but this process is quite different from those. Here we will provide monoclonal antibody against the cancer cell. Before going to talk about the main process, we need to know a basic property of cancer cell, right? In normal cell, there are various growth factor receptors, suppose epithelial growth factor receptors, but they are present in normal cell in small amount. They are helping in development and growing of the normal cell, right? And here MHC class 1 molecule are present in sufficient amount that can be recognized by a TCR, right? But when this normal cell somehow converted into a cancer cell, this may be a cause of mutation, this may be a cause of viral infection, whatever. It will converted into a cancer cell, and when it will converted into a cancer cell, it will immediately reduces the expression of MHC class 1 molecule, and that is a way by which cancer cell, which also we call them as tumor cell, evades our immune response, right? Because as MHC class 1 is in lower expression, TCR cannot recognize the cancer cell well. So if TCR cannot recognize, TH will not be activated, and TH if not activated, it will in turn not activate the TC cell. So overall immune response will collapse. That is why cancer cell have the ability to evade our immune response, because they have the ability to lower the expression of MHC class 1 molecule, but in turn they will increase the expression of epithelial growth factor receptor 2. That is why, and this epithelial growth factor receptor, when binds with a epithelial growth factor, then it will induce some signaling pathway inside that will provide a signal to the cancer cell to rapidly divide and grow, right? That is why cancer cell is rapidly dividing and growing. It is also a main cause, and we are going to produce monoclonal antibody against this epithelial growth factor receptor for our cancer treatment. So let's see how. For doing this, we will use a rat or rabbit for producing monoclonal antibody, right? This monoclonal antibody will have specificity towards these receptors, epithelial growth factor receptors, right? So when we inject this monoclonal antibody, this monoclonal antibodies will go against this epithelial growth factor receptor and marks the epithelial growth factor receptor, so that this epithelial growth factor cannot bind with this epithelial growth factor receptor. That is why the signaling pathway will not started. That is why cancer cell will not able to grow further and divide, right? In that way, we can provide our cancer treatment by simply making a monoclonal antibody that have the specificity towards epithelial growth factor receptor, and they help to they help to mark this epithelial growth factor receptor against epithelial growth factor, so cancer cell then not able to grow further rapidly, right? And it will start acting it is quite like our normal cells, right? As they will lose one of the main property of them, that is rapidly growing and dividing. Now there is also a problem. So what is the problem? That is, we use rat and rabbit to produce our monoclonal antibody, right? So if we inject or provide this into our human body, the human body will in turn provide activate their immune response against this FC region, right? As it is coming from a rat or rabbit, it will obviously show allotypic the immune response, right? Because it is coming from a completely, it is not only allotype, it is also so yeah, isotypic immune response too, because it is coming from a completely different species. That is why it is also a immunogen, right? So a antibody produced from this rat or rabbit have the ability to induce our own immune response, so that can be a problem. But so as a solution, we will prepare the FC region with the help of human antibodies, right? We can do this by help of a process called chimerization. This chimerization is a process of hybridization, and this is a process of genetic engineering by which we have the ability to produce an antibody of which FC region is from FAB region. This FAB region is from a one species, and FC region is completely from another species. That is why we will produce a antibody whose FC region is from human and FAB region is from mice. So our problem will solved, and in that way we can treat our cancer cells, right? One example of this is breast cancer, where the receptor is HER, and antibody that we will use in that case is Herceptin. Not only we provide the monoclonal antibody, right, but we also have the nowadays we also conjugate a type of immunotoxin with them. This immunotoxin have the ability to create a pore in the cancer cell or causes destruction. That is why we also conjugate this monoclonal antibody with a kind of immunotoxin. That what I have written here. Monoclonal antibodies sometimes also conjugated with toxin that can be dangerous for the cancer cell. So the overall process is, here our cancer cell, we will produce a monoclonal antibody whose FC region, whose FAB region is coming from rat or rabbit, showing specificity towards epithelial growth factor receptor, and marks our epithelial growth factor receptor from epithelial growth factor, and FC region is from human, so that the patient's body will not in turn produce a immune response against this antibody. In that way, we can treat the cancer by using monoclonal antibodies, right? So hope you will like the video. Hope So hope you like the video, and if you like the video, please hit the like button, share it with your friends, and subscribe our channel. Thank you.